Okay, hi, good afternoon, everyone. I'd like to begin by acknowledging the traditional owners on which land we meet today, the Wurundjeri people of the Kulin Nation, and pay my respects to elders past and present, and those that may be joining us today. So it is my great pleasure to introduce Jin Chan today for her completion seminar. And so I'm going to tell you about her journey. She did her honors with us in 2016, and she was basically at the end of honors. I was really keen for her to join us as a PhD student. And I remember having this discussion with her about what she would like to do. And she mentioned that she would really like to be a structural biologist, which I was extremely pleased about. But as most of you know, I myself am not a structural biologist. And so I had to basically lead from a place of ignorance. And I want to just thank Mike Lawrence for always joining us on this journey, and for a lot of her mentors as postdoctoral fellows, Melanie, Phil, Lynn, for helping her on this. Legion's PhD projects has really touched some of the most exciting parts of our program. They have been really exhilarating programs that we've done. She led a lot of the biology around of the Vivax program around RBP2B, trying to understand those host pathogen interactions. She also joined us on the SARS-CoV-2 program, looking at the characterization of nanobodies as well. And then finally, she's joined us on our future journey with Melanie on 16 proteins that she won't have time to talk to you about, but also leading on those characterization of those nanobodies against 16 programs, which is our plan for the next 5 years. She is an incredibly deep thinker. She is an exceptional experimentalist. And everything she touches is just, she's got these amazing green hands. And I just have appreciated so much time to be able to talk to her in these last couple of years. She's just this incredible person to discuss science with. And I hope you enjoy her talk. And I very much look forward to fielding your questions. Thanks.
Thanks for that introduction, Waihong. So today I want to start with this slide showing the death toll caused by the major pandemics in our history, which highlights the major impact that infectious diseases can have on global health. As one example, the 1918 influenza pandemic, which was beginning just as World War I was ending, killed more people than in the war itself. And even in the absence of pandemics, still 30% of global death and disability burden is due to communicable diseases. So today I wanted to talk about antibody therapeutics, as they have the potential to decrease the global burden due to infectious diseases, as well as tackle emerging infectious disease outbreaks. The antibodies are naturally developed by the immune system to fight disease, and they do this by binding like lock and key to a body's foreign invaders. And the discovery of the existence of antibodies followed quickly with its use as a therapeutic to treat diphtheria, which is a major cause of childhood mortality at the time. In 1890, Bering and Kitasato determined that the serum from diphtheria-infected animals could be used to treat other infected animals. And this was later found to be effective also to treat humans. They called the active ingredient in the serum antitoxin, which we know as antibodies. And the serum therapy has been used to treat some bacterial and viral infections since then. Many people have contributed to our understanding of antibodies and the immune system, including a former WEHI director, McFarlane Burnett, who developed the clonal selection theory. And this theory states that there's a pre-existing population of immune cells that, when able to bind a specific foreign antigen, will multiply and produce antibodies to that antigen. And since then, WEHI has been majorly involved in immunology research. The next major development for antibodies as therapeutics was hybridoma technology, which enabled the culturing of single B cells, which are the antibody-producing cells, to create one clone, and these are called monoclonal antibodies. And they had advantages over the polyclonal, the pool of antibodies in serum, and that you could select your most potent antibody to be used as a treatment. And this led to the development of the first monoclonal antibody license for use, which was the mouse antibody. However, mouse antibodies are recognized as foreign by the human immune system, and create undesirable effects in terms of immune responses. So efforts were made to humanize these antibodies, which included putting the mouse antigen recognition domains onto a human framework. But it wasn't until the development of several other technologies, such as phage display and transgenic mice expressing human antibodies, that it was easier to express fully human antibodies. And this led to the licensing of the first 2 fully human antibodies in the 2000s. And going forward, antibody therapeutics will most likely be fully human antibodies. And monoclonal antibodies play an important role alongside vaccines in protecting populations from infectious diseases. Whereas vaccines take time to generate an immune response, antibodies can be administered directly to treat the disease. And where vaccines are effective in individuals with functional immune system, monoclonal antibodies have a space in treating immunocompromised populations. And antibody therapeutics has seen massive growth over the past few years, and there are currently more than 100 that have been FDA approved for use. And in 2019, 7 of the top 10 best-selling drugs were antibody therapeutics, and these were for the treatment of autoimmune diseases and for cancer. And despite this over 100 antibodies being approved, only 7 have been approved for infectious disease treatments, which are indicated by the blue bars on this graph. As you can see also by the green bars, there are several antibodies that are in development, either in preclinical or clinical trials. So 3 antibodies are used for the treatment of bacterial infectious diseases and 4 for viral infectious diseases. And those include for the prophylaxis of respiratory syncytial virus, 2 are used for the treatment of Ebola virus to reduce mortality, and 1 for HIV. And to highlight some of the advancements and advantages in using antibody therapeutics to infectious diseases, I wanted to use HIV here as an example. And this field of research has really led the work of antibody therapeutic development to infectious diseases. So HIV has more than 60 strains, which makes targeting the viral antigens difficult because of that diversity. However, in the 1990s, researchers discovered the first generation of broadly neutralizing antibodies, and they created these antibodies through phage display and hybridoma technology. Those antibodies are shown in the table here, targeting these colored domains on the viral envelope protein. And they showed moderate efficacy in viral neutralization assays, as well as a broad range of efficacy when it came to neutralizing different strains, which is indicated by coverage. It wasn't until the development of single B cell approaches and high throughput neutralization assays that the second generation of broadly neutralizing antibodies were in development. And this allowed a screening of a broader pool of antibodies, which led to the isolation of a much larger number of neutralizing antibodies, as well as uncovering new sites of binding by neutralizing antibodies to the viral envelope protein. And these antibodies showed higher potency as well as higher coverage. And as well as single antibodies being able to target multiple strains of the virus, multiple antibodies can be used in combination to decrease the chances of immune escape mutations that would decrease antibody potency. And this is one of the major advantages of using antibodies in targeting the viral antigens in different ways. Again, here in the middle is shown the example for HIV, bound by these colorful antibodies. On the left here is actually the approved Ebola antibody therapeutic Inmazeb, which is a 3-antibody cocktail, and you can see these antibodies bind multiple sites on the viral protein. And even for COVID, we now have a good idea of the landscape of neutralizing antibodies binding. As we have seen, several variants have emerged through the pandemic, and this combination antibody approach will most likely be effective in targeting these variants. So in my PhD, I looked at antibodies towards 2 of the most deadly human diseases, malaria and COVID-19. And I'll start my talk with what I've done in malaria research. So malaria is caused by the Plasmodium species, of which there are 6 that are able to infect humans. And in my PhD, I worked on Plasmodium vivax. And the incidence of vivax in 2017 is shown globally on this map here in blue. In 2017, there were an estimated 14.3 million cases of vivax and 3.3 billion people still living at risk of the disease. Vivax, as well as Ovale, have another hurdle in terms of eradication, in that they can create dormant liver forms that later on cause relapses in disease. This is the life cycle of Plasmodium vivax, and it has developmental stages in the mosquito as well as the human. But today I'll be focusing on this red blood cell cycle which occurs within the human. The parasite at this stage is called a merozoite, and it invades red blood cells, replicates within them to produce new merozoites, then can rupture the cell and go on to invade healthy new red blood cells. And this is where the clinical symptoms of malaria arises. Antibodies can be used to target the parasite at this stage and prevent it from entering the host cell, but we need to understand the host pathogen interactions that occur to be able to target these. And for vivax, its host cell are reticulocytes, which are young red blood cells. And they differ from mature red blood cells. Well, they can be distinguished by the presence of this reticulum, which can be stained, and here is shown in blue. So there's differences in the receptors displayed between reticulocytes and mature red blood cells that enables vivax to specifically recognize the reticulocyte host cell. The first antigen that was identified for vivax was Duffy antigen. And this was through the observation that a closely related human malaria species, Plasmodium knowlesi, was unable to infect Duffy negative red blood cells. And this phenotype is well described in Africa, where vivax prevalence is low. And so later, Duffy was also identified as a receptor for vivax. However, Duffy is present on both mature red blood cells and reticulocytes, so it is not a reticulocyte-specific receptor. And that was a long-standing question in the field of vivax. What is that reticulocyte-specific receptor that enables vivax recognition of its host cell? This is what we know about the host pathogen interactions between falciparum, which is considered the deadliest malaria parasite species, and vivax. There are 2 main families of proteins that are involved in this specific interactions with host cell receptors. And those are the erythrocyte binding proteins, which are the EVAs in falciparum and DBP and EVP in vivax, and the reticulocyte binding protein family, which is the RHs in falciparum and RBPs in vivax. And in vivax, for a long time, the only interaction that was known was between DBP and Duffy antigen. However, since then, 2 other receptors have been identified, transferrin receptor 1, which binds to 2B, which was identified in the TALM lab, and recently CD98 was found to bind 2A. And both of these are reticulocyte-specific receptors and can govern vivax recognition of its host cell. So here again are those receptors known for vivax, and in my PhD, I worked on 2B binding to transferrin receptor 1. And in this paper, we showed that JK1 cells, which are a red blood cell line, expressing a mutant version of transferrin receptor 1 were refractory to vivax invasion, but not to wild type. Whereas falciparum had no difference between invasion of transferrin receptor 1 mutant or wild type cells. And this indicates that transferrin receptor 1 is critical for Vivax invasion. Transferrin receptor 1, which is also known as CD71, is involved in the iron regulation in humans. And it does this in complex with several human ligands. So here's the transferrin receptor 1. It has this butterfly shape. It can bind to transferrin, shown here in orange, which is an iron carrier. It can bind to HFE, which regulates the binding of transferrin. And it can also bind this heavy chain ferritin, which is an iron storage molecule. Transferrin receptor 1 is also co-opted by several pathogens as an entry receptor. Here, as I've mentioned, 2B is able to bind transferrin receptor 1, as well as the New World Hemorrhagic Fever arenavirus, virus, and its GP1 protein shown here in pink. And it's also an entry receptor for hepatitis C virus. So this brings me to my first aim, which is to perform structure-function analysis of the 2B transferrin receptor 1 interaction site. So as I showed on the previous slide, we know what this interaction looks like with the cryo-EM structure. So here in purple is 2B binding, 2 molecules of 2B binding to transferrin receptor 1 in green. And this complex reveals that 2B also binds a smaller site at the bottom here to transferrin. So what I did in my PhD was to express mutants of 2B where residues that were involved by the structure implicated in forming salt bridge interactions, shown here in red, and some hydrogen bonds in green, were mutated to alanine to identify the contribution of each of these interactions to complex formation. The assay I used to look at this was a reticulocyte binding assay, where we could detect 2B binding to transferrin receptor 1 naturally present on reticulocytes. Wild type was here set to 100%. As you can see, there are 2 mutations, tyrosine 542 and lysine 600, where we see no 2B binding to reticulocytes. The next assay that I used was an analytical size exclusion chromatography. So the wild type recombinant proteins are able to form a complex and elute a little after 10 mil, whereas this deletion in glycine 217 of transferrin receptor 1 we have previously shown doesn't bind to 2B, and the separate complexes would elute at about 11.4 mil. And again, lysine 600 and tyrosine 542 don't show complex formation by this assay. So taking a closer look at these interactions, tyrosine 542 forms hydrogen bond interactions with the backbone of transferrin receptor 1, but more importantly is part of this 3-tyrosine motif, which forms pi-pi stacking interactions with tyrosine 211 of transferrin receptor 1. Lysine 600 is involved in salt bridge interactions. And this arginine 359 mutation, although I didn't show it in the previous slide, I wasn't able to express an alanine mutant at this point, but alanine mutation of the reciprocal residue on transferrin receptor 1, glutamate 149, also showed no complex formation. So we know this residue is also essential. And as you can see, all these residues are at the transferrin receptor 1 interaction site. So in terms of isolating an inhibitory antibody, this site is probably what we would want to target. As an interesting side note, as I mentioned, machupavirus GP1 also binds to transferrin receptor 1 and does so contacting the same sites as 2B. And both of these pathogens rely on this tyrosine 211 for complex formation. In machupavirus GP1, this tyrosine forms a hydrogen bond, but also binds in this pocket created by these residues. And tyrosine 211 is a residue that determines host tropism of New World Hemorrhagic Fever arenaviruses. So it is interesting how these 2 different pathogens have co-opted the same receptor and also the same residue for binding and complex formation. Finishing up with that structure functional analysis, I was then interested in looking at human monoclonal antibodies to 2B and performing structure-function analysis of these antibodies. So my work with antibodies to 2B began in my honours, where I characterized mouse monoclonal antibodies that were able to inhibit 2B binding to reticulocytes. The reticulocyte binding assay is shown here by these representative dot plots, where reticulocytes are separated on the x-axis and the binding of 2B to the reticulocytes is shown here on the y-axis. An addition of an inhibitory antibody such as 3E9, we see a reduction in this binding. We tested these antibodies in short-term Vivax invasion assays, and they showed about a 50% decrease in Vivax invasion efficiency. So leading on from this work, I was interested in looking at human antibodies and how they would behave. And we know that from studies that in Vivax endemic populations, that antibodies to 2B are generated, as well as to the other RBP members of Vivax. In this high transmission setting of Papua New Guinea, antibodies to 2B as well as 1A are associated with a reduced incidence of Vivax malaria. Whereas in Brazil and Thailand, where Vivax transmission is low, only antibodies to 2B are associated with protection against Vivax malaria. So from here, we collaborated with the King Lab at Case Western Reserve University to isolate naturally acquired human monoclonal antibodies to 2B. We did this through single B cell sorting, and through this technology, we isolated 300 2B specific B cells from 2 Cambodian individuals with a history of malaria infection. The antibody clonal groups are shown here for these 2 individuals, and in my PhD, I expressed 1 to 2 antibodies from each of these clonal groups to characterize in my assays. And in total, I expressed 23 antibodies. So these are the 3 assays that I used to look at the function of these antibodies. I've gone through the reticulocyte binding assay, but just quickly, we can see 2B binding to transferrin receptor 1 present naturally on reticulocytes, and that's shown here. An addition of an inhibitory antibody decreases the signal. In the FRET assay, 2B and transferrin receptor 1 recombinant proteins are labeled with 2 different fluorophores that when the complex is formed, the fluorophores are brought close together, and excitation of the 2B fluorophore causes excitation of the transferrin receptor 1 fluorophore, which can be measured here as the FRET signal. And inhibition of this complex means you don't see this FRET signal. And finally, in the immunoprecipitation assay, the antibodies are allowed to bind 2B, and this is immunoprecipitated by protein A beads and analyzed on a gel. So for a non-inhibitory antibody, we would see immunoprecipitation of the antibody, here separated in heavy and light chain, as well as 2B. And non-inhibitory antibodies can co-immunoprecipitate transferrin receptor 1 and transferrin, whereas for an inhibitory antibody, we don't see transferrin receptor 1 and transferrin. And as you can see, there are some differences in all 3 of these assays, which means some of the antibodies behave differently in these different assays. So this is the result for my reticulocyte binding assay. I've summarized the results in this colored table up here, where green are inhibitory antibodies, yellow are intermediately inhibitory, orange not inhibitory, and an enhancing antibody. The results of my FRET assay are also summarized here, with green being inhibitory, orange not inhibitory, and 1 enhancing antibody again. And for the immunoprecipitation assay, the same color coding with inhibitory in green and non-inhibitory in orange. And as I mentioned, there are some differences in the assays for some of these antibodies, but we do see 8 antibodies that show consistent inhibition across all 3 assays. I also measured the affinity of these antibodies and all of these by bilayer interferometry, And all of these antibodies were quite high affinity, the lowest being 0.9 nanomolar, which is still quite high. But what do these affinities mean in relation to the inhibitory potential of these antibodies? To look at this, I've plotted here on the x-axis the affinity of the antibody, and on the y-axis the inhibition by the reticulocyte binding assay. And as you can see, the inhibitory antibodies in this reticulocyte binding assays have a range of affinities. And in summary, a KD of less than 1 nanomolar for these antibodies is still sufficient to see inhibition. From here, I was interested in looking at where these antibodies bound so that we could suggest a combination of antibodies that can be used to target 2B. We did show that most of these antibodies bind to this domain that mostly forms interactions with transferrin, and 5 were isolated that bound this top region here. Anu from Chris King's lab did a competition assay. So in this table, blue squares indicate where antibodies are competing with each other for binding to 2B. And as you can see, there's in general 4 main groups, 4 locations where antibodies are binding. And in theory, you could choose one antibody from each of these groups to target the 2B protein. To more finely map where these antibodies are binding, I used X-ray crystallography and determined the crystal structures of 8 antibody Fab fragments binding to 2B, shown, and I've superimposed their structures here. So in this image, the region of 2B that was able to crystallize is colored here in gray, and I've superimposed it on the cryo-EM structure. So breaking these structures down, there are 4 antibodies that directly bind at the transferrin receptor 1 transferrin interaction site, and these consistently show inhibition across all 3 assays that I used. This one antibody seems to disrupt transferrin binding, and it has some different effects in the different assays. And 3 antibodies were binding opposite to the transferrin receptor 1 transferrin interface. And this antibody didn't show any inhibition. And these 2 antibodies did, for the most part, didn't show inhibition, except in the red blood cell binding assay, they did show some inhibitory potential. And I'll come back to why that might be in the next few slides. We were still interested, so I was only able to obtain crystal structures for 8 human antibodies out of 23, but I was still interested in mapping where these other antibodies were binding. And to do this, we collaborated with Dominique's lab at the University of Geneva to do hydrogen deuterium exchange mass spectrometry. And how this technique works is that you have your protein in solution, and the protein has hydrogen atoms that, when in solution, can exchange with the hydrogens in water molecules. When you put this protein into a deuterium solution, then deuterium starts to exchange with the hydrogen atom on the protein, and this change can be tracked. And this only happens when to surface exposed sites. So when you add your antibody, the antibody would bind to a region and shield that region from the solution, resulting in a decrease in that exchange. And this difference between hydrogen deuterium exchange in the presence or absence of antibodies can generally map where the antibody is binding. So I did this for, at the University of Geneva, they did this for all 23 antibodies, but I'll summarize just the most important findings. So for 4 antibodies, we were able to look at their crystal structures and see how they were able to inhibit across all 3 interaction assays. For the remaining 4 antibodies, we were able to map these epitopes using hydrogen deuterium exchange mass spectrometry. So 2 of these antibodies are binding at the tip of the 2B protein, and the other 2 binding onto the other end where transferrin receptor 1 interaction site is. And hydrogen deuterium exchange also gives us an explanation of how these 2 antibodies, which don't seem to be inhibitory according to their crystal structure, may have some inhibitory effects in the reticulocyte binding assay. And that is because here I've superimposed the crystal structure onto the region that is affected by hydrogen deuterium exchange, shown in blue. As you can see, the binding of this antibody does cause some changes in this loop that threads in between the 2B structure, comes out the other side to contact transferrin. So the changes in this loop could be causing this decrease in 2B binding to transferrin receptor 1. From these mapping studies, I can propose a combination of antibodies that can be used to target 2B. That would be 3 antibodies that show inhibition in the reticulocyte binding assay. And it would be interesting to see how this antibody binding opposite the transferrin receptor 1 interaction site would affect Vivax invasion. So to summarize this part of the talk, I've characterized the transferrin receptor 1 2B interface and identified critical residues for complex formation. I've also characterized naturally acquired human monoclonal antibodies to 2B and found several that inhibit complex formation. And I also propose a combination of antibodies that can be used to simultaneously bind 2B. So moving on to what I have done in terms of COVID-19 research. So COVID-19 probably doesn't really need an introduction, but just some stats on the disease. To date, there are 190 million confirmed cases and over 4 million deaths. And over the course of the pandemic, several variants of concern have arisen. So what is the host pathogen interaction that we can target in terms of SARS-CoV-2? That would be the spike protein shown here, which is involved in attachment to host cells and entry. And this is through the receptor binding domain here, which contacts the human receptor, which is ACE2. And it can only do this in the open conformation. So as I was wrapping up my project with the 2B human monoclonal antibodies, the pandemic hit, and the TARM lab started characterizing nanobodies produced in alpacas to that RBD domain of SARS-CoV-2. It was here that I took a 6-month break from my PhD to work as a research assistant and characterize these antibodies and looking at measuring their affinities and doing competition assays to see which nanobodies could be used in combination. And continuing on from this work, in the last 6 months of my PhD, I attempted to do affinity maturation on one of our lead SARS-CoV-2 antibody candidates in order to improve its potency. So coming from this paper that we published this year, we characterized 52 nanobodies targeting that receptor binding domain and identified lead candidates by the potency in viral neutralization assays, as well as the highest affinity nanobodies. And from this, we obtained 4 lead candidates. These nanobodies were able to neutralize the N501Y variant of the virus, as shown here by the colored lines, compared to the isotype control, shown here in black. And when administered prophylactically to mice, they were also able to prevent viral infection compared to the isotype control shown here in black. So looking at these curves, WMB36 was our best performing nanobody in this assay and was the one that I used for affinity maturation studies. Which brings me to my last aim, which was to generate a high affinity variant of WMB36. So to do this affinity maturation, our approach was to randomly mutate WMB36, create a large randomly mutated library, and then select for high affinity variants. To do this, I used the Multiplex Assay Technology Hub, or MAP platform, which is a WeHi initiative led by Melissa Cole and Alan Rubin. This platform uses deep mutational scanning technology, and what that is allows us to select any position along our nanobody sequence and mutate that residue into all the 20 possible amino acid residues plus the stop codon. And we did this for 36 across the whole nanobody sequence. And to further increase diversity, we introduced multiple mutations at the same time. And an idea of what we want to achieve is shown in this figure here, where in this paper, they were able to identify several residues highlighted by the spheres, which increased thermal stability or increased affinity, or both. So this is the general workflow of how we would conduct these affinity maturation experiments. First is to generate the large randomly mutated library, and for 36, I obtained a library size of 4 × 10⁷. Then to display these to express these nanobody variants, I use phage display, where the phage is able to express the nanobody on the surface. We then expose the phage to our receptor binding domain to select for phage that are able to bind, and this is called panning. And we did this panning 3 times, each time decreasing the amount of antigen so that we're left with high affinity nanobody variants that can outcompete the lower affinity variants. And in this way, we can enrich for higher affinity nanobodies. To identify the nanobody variants, we attach a barcode, and the frequency or the enrichment of the barcode can be determined with Illumina sequencing. We can then identify the nanobody sequence attached to that barcode with PacBio sequencing. And once we have our nanobody variant sequence, we can express these and test them in our assays. And I'll be using the FET assay to look whether they retain their ability to inhibit the complex of RBD and ACE2, and measure their affinities and thermal stabilities. This is the first time we have used the math platform to conduct affinity maturation experiments, and I unfortunately, due to time constraints, wasn't able to pull out the nanobody variant sequences of the enriched barcodes, but that is that information will be available to the lab. So this is the Illumina sequencing data, essentially showing that we see enrichment during our panning stages. So the barcodes are shown on the number of barcodes are shown on the y-axis, and the number of times they have been counted on the x-axis. And this is shown for before pannings, pan 1, pan 2, and pan 3. And as you can see, looking at the tail end of these graphs, there are an increasing number of barcodes that are counted an increasing number of times. And this indicates that we have seen enrichment during our panning process. And these enriched barcodes are the nanobody variants that we would be interested in. But as I mentioned, I didn't have time to pull out these nanobody sequences. So instead, I chose a random selection of nanobodies from pans 1, 2, and 3, looked at the binding to the receptor binding domain by ELISA, and chose top 7 nanobodies in terms of signal from each of these pans to express and look at in terms of my FRET and affinity and thermal stability assays. Because these number of nanobodies is relatively small, we were able to sequence all these nanobodies to create a heat map looking at mutational frequencies. So on the x-axis here is the nanobody sequence, and on the y-axis, all the possible amino acid mutations and the stop codon. And the red to white color scale indicates the number of times a particular mutation has been counted, and gray indicates no mutation was counted. No mutation of that sort was observed. If we were creating this heat map from the enriched sequences, we would expect that high frequency mutations are correlated with an increase in affinity, because that is the property that we are selecting for. However, since I chose a random assortment of nanobodies to analyze, this map doesn't show us such information. What it does show us, interestingly, is that there are 12 positions that have no mutations. For example, this cysteine at position 50, which is gray all the way down. I mapped these 12, or highlighted these 12 residues on this structure of WMB2, which is closely related to WMB36. And essentially, you can see these residues are involved in disulfide bonds. This one is most likely important in positioning the CDR3 loop. There's also the salt bridge interaction, but most are hydrophobic residues that are pointed towards the core of the nanobody structure. And all these residues are most likely important for nanobody folding and structure. So in future deep mutational scanning attempts, we may want to exclude these residues when we do the deep mutational library. So coming back to the nanobodies that I expressed, of the 20 ones that I attempted to express, only 11 showed any nanobodies. And these are SDS page gels of the crude extracts, where nanobody is running at a little less than 15 kilodaltons. And I was only able to purify 7 nanobodies with enough material to perform experiments, and those nanobodies are shown on this gel. I also included the size exclusion chromatogram just to show that some of the nanobody variants did run a bit differently from wild type, which is shown by the black line. So the first assay that I used was the FRET, and only 1 nanobody lost the ability to inhibit this complex formation. And that's most likely because P2H5 did show precipitation in solution. So we're probably not adding as much nanobody as I thought I was. I measured the affinities, and while I didn't see any increases in nanobody affinities, I did see 3 nanobodies that decreased in affinity compared to wild type. And in terms of thermal stability, because these gray lines on the graph indicate where wild type nanobody, the melting temperature of wild type nanobody, and the green line for the variants. And while I didn't see any increases in thermal stability, 2 variants did show a decrease in thermal stability. And I didn't have time to show I do have structures showing where these, suggesting why we see these phenotypes, but I haven't had time to show them today. So in summary, I was able to create a WMB36 variant library using the Math Deep Mutational Scanning platform. While I wasn't able to identify higher affinity or higher thermal stable variants, I did identify residues that are important for nanobody structure. And I've also established a pipeline for testing future nanobody variants. And as Waihong mentioned, I was also involved in other projects, including the characterization of nanobodies to 6-cysteine proteins by measuring their affinities and conducting competition experiments. And we have a published paper for that. So that leads me to my acknowledgements, which I probably don't have time to go through as well as I would like to. But first, I would like to thank Waihong, my supervisor, for all the opportunities she's provided and just her support. And I think she's believed in me more than I've believed in myself during my whole research career. I'd like to thank everyone in the Town Lab, past and present, especially Jacob, who taught me a lot about structural biology at the start, and Melanie, who, as soon as she joined the lab, quickly took over that mentoring role. And as everyone in the lab has contributed to what I know in terms of various experiments here and there. In particular, thanks to Matt for helping me with the WMB36 library, and Damien for showing me how to do the phage display panning. I'd also like to thank my PhD committee, who at various times during my PhD have provided advice and answered my questions regarding all different sorts of parts of my project. I would like to thank the WeHi Math platform, in particular Melissa Cole and Margareta Gove, for teaching me the deep mutational scanning technology. I'd also like to thank Stephen from the Kalman Lab for answering all my questions related to octet, as well as the Bandura team for always welcoming me up to Bandura for my long experiments using the octet. Also, everyone in the ID2 division, it's been an amazing place to work. I would also like to thank our collaborators from the Case Western Reserve University, as well as the University of Geneva, as well as my family and friends and my partner. And thank you, and I'll take questions.
Great. Thank you so much, Jin, for such a wonderful talk. Just wanted to say that, as I mentioned in the beginning, she came at the end of honors to say she wanted to be a structural biologist. And it's been incredible to hear the story where you've solved, very easily 8 human monoclonal structures. And she, during her RA-ship working on the COVID program, solved another 2 to 3 nanobody structures against SARS-2 RBD, and just, really attribute to her tenacity as a scientist. And what I wanted to say in my introduction is just the courage that she's shown through her PhD to take on a skill that her supervisor has no clue about, and just been an amazing scientist coming through. So congratulations, Jin, on a wonderful completion seminar. So we have the first question by Matt Cole. And Jin, Matt says, fantastic work and thanks for a great talk. I realize that these aren't therapeutically useful in terms of the RBP2B antibodies, but how do you explain your 2B transferrin receptor enhancing antibodies? Any ideas?
Yeah, I think we did see that one antibody 2792280. It's been a while since I've said that, but that did show enhancement in the reticulocyte binding assay as well as the FRET. In terms of, I think we can kind of hypothesize how this antibody is increasing that interaction between 2B and transferrin receptor 1 by where it's binding. And by HDXMS, we see that it does bind opposite the face of the transferrin receptor 1 binding site on that stem of 2B. So I think what's happening is that antibody binding is locking 2B into a conformation that is favorable for its association with transferrin receptor 1, and that's why we see that enhancement.
Great. And you've got a question from Chris. Chris says, great talk. Does 2B bind transferrin receptor without the C-terminal domain? If so, why does the tyrosine mutation in this region prevent binding of the longer protein?
That is interesting. So the fragment that we initially found to bind transferrin receptor 1 was that N-terminal domain alone. So we do know that that domain itself can bind to transferrin receptor 1 and transferrin. I think with the longer structure, if you remove the critical contacts between that longer fragment, because it is a bigger protein, it would cause more destabilization, and the hydrogen bond, well, the transferrin interaction site is only made up of hydrogen bonds, and that might not be enough to stabilize the longer complex. So I think, if you do have that extended N-terminal domain, it does require these salt bridge interactions and the pi-pi interactions to really stabilize that complex.
Great, thank you, Jin. Well, we're waiting for questions. I do encourage people to put in more questions in the chat. But Jin, just coming out, I know you didn't have time to talk about this in terms of the MAT platform, but when you did the deep mutational scanning, were there particular mutations that you couldn't get? And is there a different way that you would have potentially approached the deep mutational scanning moving forward?
By mutations that we didn't get, do you mean we did see 12 positions where we didn't get mutations? I'm not sure if that's what.
Yeah, that's what I mean. And could you talk a bit about them? Yeah.
Yeah, I think as I showed on that structure of the nanobody, that these residues are important for the nanobody folding and its ability to fold into a functional nanobody that can bind the receptor binding domain during the panning stage. So mutation of these residues could lead to unfolding of the nanobody, and then we don't actually see that nanobody progressing through the panning stages. And then in terms of different ways we can approach the deep mutational scanning, I think one of the really interesting ways we can approach this is we do have a crystal structure of WMB36 binding to RBD, which I haven't included in my talk, but it was solved recently by Phil. And using that structure, we can identify residues that are of WMB36 that are interacting with the RBD and target these residues for the deep mutational scanning. So it would be a more targeted and a structure-guided approach to obtain affinity matured nanobodies. But the reason why we chose to mutate the whole nanobody sequence is that we're also interested in looking at mutations that affect stability, and those are most likely along the whole nanobody sequence.
And Melissa Core has a question, Jin. Before we had vaccines for COVID, there was a concern that a vaccine may enhance disease. Have you observed any nanobodies that enhance the RBD ACE2 binding?
Yeah, that's a really important and interesting point, and definitely a concern when it comes to vaccines and the polyclonal responses that they can create. From what I understand, the observation of antibody-dependent enhancement of infection, which is also seen in other respiratory viruses, including COVID-19, as well as Ebola, and even some papers have observed that for malaria. And that antibody-dependent enhancement of invasion of entry is infection is most likely due to the FC-mediated interactions with the complement system. So it is hypothesized that complement proteins that bind to the FC domain of the antibodies can interact with complement receptors on cells and kind of inadvertently target the virus to its host cell for infection. So in terms of the nanobodies, we wouldn't be able to see that because we look at the nanobodies in just the antigen recognition domain. So you wouldn't have these FC related functions. But yeah, so in terms of the assays that we use, we didn't see this, and you would probably need to use a different set of assays to look at this antibody-dependent enhancement. I hope I was coherent in that response.
Great, thanks, Jin. While we're waiting for more questions to show up as well, Jin, did you want to talk to us about what you may do with these human monoclonal antibodies against Vivax moving forward?
Yeah, so I think the most important thing to show with these antibodies is that they're able to inhibit Vivax invasion. And that has been a difficulty in the Vivax field is to test these or to test any therapeutic in invasion assays because there's no long-term Vivax invasion assay. So definitely we want to see there are short-term invasion assays that we can test these antibodies on, but they require a constant source of Vivax, which is available in endemic populations. There are also several models that we could use to test these antibodies in, including humanized mouse models, non-human primates in Aotus monkeys, as well as controlled human malaria challenge models. And one other model that we could potentially adapt in the TARM lab is a transgenic model using Plasmodium knowlesi, which is most closely related human malaria species to Vivax. And it has been shown that 2B proteins can be expressed in Plasmodium knowlesi. I'm not sure if I said that before, but yeah, in Plasmodium knowlesi, and this can be used to look at the function of Vivax proteins in knowlesi, which can be cultured long term and is more easily genetically manipulated.
Thanks, Legion. So Mel says, beautiful talk, Legion, about the deep mutational scanning approach. Did I get it right that every residue gets mutated into every possible amino acid, including stop codons?
Yes.
Have you seen any shorter nanobodies that were still able to bind?
Yes. So in terms of the nanobody platform, stop codons are read as are not actually coded as stop codons in the phage display. When the phage infect E. coli cells, specialized E. coli cells, they can actually be, stop codons can actually be coded as a glutamine, specifically TGA stop codons. So the stop codons in our panning are actually coded as glutamine, and that's something that I checked in the 384 random sequences I pulled out, that stop codons were actually read through. But when you express these nanobodies in an E. coli strain that doesn't have this amber suppressor kind of phenotype, it would code a stop codon. Yeah, so in our panning stages, we do express them. We don't see any shortage of nanobodies, but when we express them as actual nanobodies in culture, they would produce stop codons, which is something that we had to. So we had to, for these nanobodies that we saw a mutation of the stop codon, we had to order a synthetic gene to actually code for a full-length nanobody.
Great, thank you, Legion. And we're smack bang at 4 p.m. We've got one more question from Phil, which we can answer separately later. But yeah, thank you everybody for being online, and in particular, if Legion's family and friends are on here, I just want to say a personal thank you to all of you for supporting her through her PhD career. And we just wish that we had been able to meet you in person today, but we look forward to catching up with everyone, hopefully as we come out of the pandemic in future weeks. But just take a moment for me to just basically thank Legion and in giving a wonderful seminar today.
Thank you, everyone.